Good evening. For those of you who don't know me, I'm Tim O'Shea. I'm the Principal and Vice Chancellor of the University. A tremendous pleasure to welcome you to Professor Vincent Danos' inaugural in his role as Chair of Computational Systems Biology. So we're about 2 years late? Yeah.
About
2 years late. Very pleased that his wife, Dr. Ellen Feshavi, is here. It is very important for partners to support our partners as they are doing inaugural lectures, because these are important combined academic and family events. Vincent was trained at university level in the École Polytechnique in differential geometry. He then was an ingénieur du corps du pont et chaussée. Right.
That's right.
Which means he was being en route to becoming an elite civil servant. I'm sure he was sympathetic to bridges, but he didn't actually do anything with bridges or roads.
But bridges are a powerful metaphor, right? So.
And then he changed direction a bit, did a PhD in mathematics, focusing on logic, moved to the CNRS, which is the famous French research institution, and as we all do, moving from things like the civil service or the private sector to the world of the university. In his case, he suffered a 50% cut in salary. But we do this joyfully for the intellectual freedom that we acquire as we make these moves. And it's really quite strangely, from my point of view, as somebody who was a very young computer scientist, he only got into programming then, learning functional languages at the CNRS, and he also got interested in distributed systems, which took him into his biological interests. So he is a very youthful, if I can put it this way, computer scientist and a very youthful systems biologist, but an extremely successful one. He became a Director of Research at CNRS in 2004, spent time at Harvard, developed an immensely strong international profile, and very happily joined us in 2008 here. He is triple-hatted. He's a member of the Informatics Life Sciences Institute. He's a member of the Laboratory for the Foundations of Computer Science. And very happily, he combines this with the role as Director of Research at CNRS for the Équipe Preuves program Systèmes. Is that right?
That's right.
And so this is a very, as a deeply international university, to have an equity share in a computational systems biologist who also, CNRS has an equity share, and I think this is extremely good for both sides. And he's going to address us tonight on the topic information carriers in biomolecular networks. Please.
Thank you very much. Thank you for this very nice introduction. So that lecture will be about biomolecular networks, which means networks of interacting molecules coming from the actual stock of biological systems. And the stress of the talk is about, how are these networks actually computing? What kind of principles are governing their local interactions? So how do the biochemical components actually do what they do, and how do they bind together? How are they complementary to one another? How do they modify one another? That's the local aspect of the network. And then, of course, there is the global network aspect of the network, which is to bring about some sort of consistent behavior, macroscopically reliable. So these are the questions I'm going to get into. But before I do that, and because I'm a logician, essentially, I would like to get into the problem of which language we're actually going to use to do this. And the reason I'm putting this to the forefront is that I think language matters. So in a modeling situation, language matters in an obvious way, because without a language, you're not going to be, without a syntax, like a machine-readable language, you're not actually going to be able to put together a description of your system that you can set into numerical motion on a computer. So that's an obvious way in which languages are useful. But they're also useful in another way. It is not every language is born equal. And when you have two descriptions of the same phenomenon of interest, then, well, sometimes one of the descriptions, despite the fact it might describe the exact same thing, the way it presents the thing can be unwieldy. It can stand in the way of understanding it. And it can actually stand in the way of a machine understanding it, which is the point here. So this is why I would like to start explaining how the language we're actually going to use came about. And so a little bit of the timeline of the language. So here it is. So when I started in the late 80s, there was a considerable interest into describing communication and distributed systems, decentralized computations with minimalistic languages. And one of the formalism at the time was meant as a sort of a low-level target for a compiler to be run on an architecture that was called the connection machine. And that was Yves Lafon's infraction nets. And the language I'm actually going to use during this lecture is very close to what he was doing at the time, with different intentions, obviously. And at about the same time, Berry and Boudel designed another low-level language to represent the time evolution of distributed systems called the chemical abstract machine. And I mention this because the chemical metaphor, so the idea that one could use chemistry and perhaps now biochemistry as a good metaphor to set up a nice, unobtrusive way to talk about the evolution of distributed systems was, I think, a major conceptual breakthrough at the time. So we fast forward a few years, and there was this really influential paper written by Walter Fontana and Leo Buss, both theoretical biologists, with strong interest into the evolutionary biology, where they suggested skimming through the programming languages literature for distributed systems and try to put them to good use to provide what the traditional dynamical system-based description could not provide. So they're making a long case, and a very interesting one, to say that traditional means that were used by biomathematicians would not be enough to cope with the systems of interest. And actually, Fontana went as far as suggesting using PyCalculus. So a few years after Aviv Regev, on a completely independent branch of knowledge growth, at the time from the Weizmann Institute, decided that actually this was a really good idea, and she did a PhD on using stochastic PyCalculus to represent biomolecular networks. So I was aware of both developments at the time, and I thought this is completely fascinating. It's a potentially unlimited a field of application for this minimalistic communication languages. And there's a quantitative aspect, and so it's both an angle that we can use because we have that culture, and it's also a challenge because we don't, I mean, in theoretical computer science, we don't have much of a dynamical system quantitative culture. So it looked like a beautiful thing to try to do. So I thought for a while, I thought for a long while, and then in 2002, I kind of came out with a first proposition, which was going to become the language I'm going to use today, and that was joint work with Cosimo Laneve. So unknown to me at the time, a team of American biochemists, theoretical biochemists at the Los Alamos National Lab, mostly Jim Fader and Bill Navacek, were designing a language that was extremely similar to mine. And it's only a few years after that I learned about their language. And finally, also at the same time, I'm coming for yet another community, still from theoretical computer science. A group at INRIA was working on a very similar formalism. So this tells you something. It tells you that there is a language here that wants to exist. And this congruence of three different attempts into a formalism that, that's about the same is maybe a good sign that you're onto something. So since then, we've been developing the tools and the means of analysis, the formal methods aspects of the language with Jean Krivine, Jérôme Ferret, Walter Fontana, who's become since then a strong collaborator, and Russ Harmer and many other people. And if I go to the present day, about a few weeks ago, there was here in Edinburgh, organized by Igor Goryanin from the School of Informatics, a huge systems biology conference called ICSB, International Conference for Systems Biology. And at that conference, there was quite a number of rule-based talks, where rule-based is kind of the generic term we use to designate this family of languages. There's a couple of others, but these are the main protagonists. And just a few weeks ago, the iGEM team that was supervised by Chris French won the Best Model Prize at the iGEM competition, which is a synthetic biology competition for undergrads that's highly competitive and very exciting, and where Edinburgh has a history of doing very well. Actually, the Edinburgh 2006 project is going to become the classic poster project of iGEM. So this year, they won the Best Model Award and a golden medal, and the Best Model Award went to a family of models that were partly based on the language I'm going to use and on, extensions. So that's for the language. So now let's talk about what the language is supposed to describe, the subject matter of the language. So these are biomolecular networks. So we're going to look at examples, but let's say right now that they seem to be an object with which science has never dealt before. There's a high multiplicity of components of different types. They interact in a very dense network of interactions. And interactions in that dense network, the complexity of that is compounded by the fact that interactions are not exclusive from one another. So there's an absolutely astonishing number of combinations that these networks seem to be, in principle, capable of. And all this is happening in a probabilistic, stochastic universe where the only driving force seems to be some sort of semi-liquid, imperfectly mixed universe. So it's really a challenge to understand how the systems can actually perform so reliably. And to me, that object, with all the challenges it brings to the table, is like the 21st century space program. It's an object of such a complexity that, so okay, it's not the space outside, it's the space within, it's the nanospace, that it will have to bring a completely new set of skills and scientists to actually make way into the difficult questions. And one of the most exciting questions, I think, is the local-global relationship, which is more or less the articulation of systems biology. So how do the local interaction means result into a concerted, meaningful behavior for the network? All right, so let's have an example. It's going to be a simple example to begin with. So how does a molecular network look like? So here, this is the yeast pheromone pathway from taken from a recent paper from Lim that was published in Science a few months ago. So this is the outside world, where on the outside of the cell membrane, and there is a chemical signal that comes in that's caught by a receptor. And somehow things will organize in a reliable fashion in a way that in the end, that particular protein, FUS3, will be activated, which means concretely chemically modified in a certain fashion. And that protein will shuttle to the nucleus and trigger some piece of the transcriptional program and elicit a response from the entire system that's well adjusted to the signal. So what are we looking at here? We have 12 proteins, including the receptor. There's an edge between any two of them if they are known or believed by experimental means to interact. And the edge is solid if the interaction is unconditional on the presence of the signal. A dashed edge means there is evidence that, for example, ST5 binds to the membrane only in the presence of the signal. So how does that simplified biomolecular network compute its response, which is to, in this particular case, to activate FUS3? Well, there are two signal paths, two ways in which the signal can flow through the system. The first one, so they both start by the same sequence of operation. We have this G protein complex here, and when the receptor actually binds the ligand, the signal, the intended signal, then ST4 will detach from this little machine and will get into an active form whereby it can actually interact with ST20 and ST5. By the way, this model has been, system, has been modeled recently by Peter Swain, also from the School of Biology, in a recent Nature paper. So I just thought I should mention this. ST5 is a scaffold, and the purpose of ST5, it's not a molecule that's active in any way, it doesn't have any chemical activity, but it brings about all the cascade of successive kinases here, ST11, 7, and first 3, at the same place. And so, and ST20 is also chemically active, so that's why they're in red. They're called kinases. I'll return to the term in a few slides. So somehow ST4 manages to both activate ST20 and to make ST20 competent to modify the internal state of ST11 in a way that it's activated as well, and to bring ST5 to the membrane in a way that everything presumably happens at the same place and is suitably amplified so that we get a reliable response. So, so what can we say here? Well, that's an example. It's simplified, but still, it's not so simple, right? There's lots of edge density here in this little network, and every precise little step is still unknown. And you know, as knowledge unfolds in systems biology, we get to know more about that mechanism. So we want to know how this thing works, and if we go one step down in the level of description, actually things get a little simpler. So this is what we're doing next slide. So these are the 11 non-receptor objects that we've been looking at, and they are decomposed as subunits that are called domains. So what's a domain? It's it's a function of the protein. So when two proteins interact, they are not actually monolithically interacting with one another. They are not black boxes. You can look at their structure. They're folding independently into subunits, and the subunits have independent properties. So this brings about a vision, and that's during the last 10 years of systems biology. It's become gradually the dominating vision of proteins as being composite objects. And if they are composite, then you can recompose them. So the way you do that, so by the way, the color code here is that an orange domain is a domain that has chemical activity, so that's susceptible to modify some kind of partner in an interaction. Whereas a green domain is the domain that's purely there to create this partnership, to make a domain-domain binding relationship. So you can think of the green domains as some sort of protein recognition code, and of the orange domains as the means whereby chemical signals are propagated in the network. I mean, you have to ask yourself, how would I, as an engineer, use this system to do things, which is properly the question of synthetic biology. So here's what they do. They split carefully in the middle all the proteins. The split points are chosen in a way that you don't cut through a domain, because you destroy the function if you do so. And then they recombine them in all possible ways. And then they take one recombination of a protein at a time, they throw it in the system, and they see what happens. So that's the paper I was referring to. And here, let's consider just one example. So this is actually one of the best examples with the most spectacular behavior. So what's happening here is that we've created a new object that was not in nature. And what we did is we took the ST4 binding site of ST5. ST5 binds ST4. So I cut off the domain that's doing this. And we took the kinase domain activity, so the orange domain of ST11, and we plugged them together. And in so doing, we also took the binding site that binds ST11 to ST20. So we have this composite object. And you know, if we believe our qualitative description, then that object is going to interact both with ST4 and ST20, and also directly with ST7. So it provides yet another way in which the signal can flow down the system. So presumably, it will amplify the response and make it make the mating behavior actually more efficient, which is what they find experimentally. So that tells you that the domain decomposition is it's not just a way of thinking. It's actually some vision of how things work on which you can base some measure of engineering. It's only a beginning, obviously, but I find it really impressive. So, so now, this was, like I said, we have about 6,000 different agent types in the system, and that's just yeast, which is reasonably simple compared to other kinds of cells. And it's a single-cell organism. And so we can take a step in scales and look at a more, from a further point of view, look at the, what the network looks like. So I'm going to impress on you 2 slides. I'm not going to spend too much time on them, just to give you a sense of which scales we're talking about here. So here's one that's extracted from a science paper 2006. It's a partial domain binding map, which is giving you this impression of a highly connected domain universe. So this is the toolkit. I mean, this is part of the toolkit you could use if you were an engineer of the systems. And every little blue pill is a domain, not a protein. So, by the way, it is well understood now that there are far more protein types than there are domain types. So technology progresses in biology by recombining existing domains, much more than it does by inventing new ones. So, so this is another map from a recent paper by Michael Tyers, also from the School of Biology, published in Science. And this is a zoom in on the detail of the kinase binding map of yeast, that same organism we're looking at. And again, the impression is that of a highly dense set of interactions. So we're back at the question we were asking at the beginning, which is how do these networks actually compute with such an edge density? So now we know a little bit more. We know that biomolecules are modular, and that modularity is based on domains. And we know that they are networked on a very large scale. So that, as a, if we apply a little bit of computational thinking to the situation, we're going to see very quickly that there is absolutely no way we can have a comprehensive view of these networks if we are to enumerate all possible combinations. It is just a sea of possibilities that we cannot start thinking of enumerating. So that leaves us 2 options, which are equally interesting. One of them is to make simple models, to forget much of what's called the background of one system of interest, and to see how far we can go with the simplified semi-mechanistic models, and to use them as something that's very similar to what people do in statistical modeling or machine learning. That's an interesting path along which you can go. And it's actually the mainstream systems biology modeling is doing exactly this. But what we want to do is we want actually to not simplify in an unprincipled way the mechanistic description and actually construct the syntactic means, that particular language I was mentioning at the beginning, to be able to give a description of the whole. What to do with the whole and with that description, that's another question. Well, I'll come to this in the next part. So that brings us to the part 2, which is the language, Kappa, that rule-based language. And the abstraction is really, we've already done it in a way, in the way we're describing things. So we're saying proteins are made of domains, fine. So I'm going to do the following. I'm going to have agents in my formal world, my formal molecular biology. They will have a name, the name of the protein they're supposed to stand for. And then I will equip them with little sites that are abstracting domains, and the sites will have both binding capabilities to reflect what's happening in the real world, and it will also have internal states so that I can talk about what's called the post-translational modifications, which are these chemical modifications that are traveling around the network. They're very simple. Now, as soon as we have an object like this, we can start talking about partial objects. So a tiny little modification of revealing a tiny bit of the structure of a biomolecule is actually going to have far-reaching consequences at the syntactic level. So here we're talking about a simplified universe where we have just 2 agent types, A and Bs. And A has 2 sites, a site called D, which is D stands here for dimerization, so the ability to self-bind. And A can bind A on D. And A can bind B on another site. And B has just 1 site, so very simple. So now I can draw an object like this. So what is this object? It says that A is bound to B, where on the site S. And it says that, well, it says absolutely nothing about D. There's a question mark. We don't know. So this is a partial object. It's not a full description of a chemical species. So we can reason by cases, and we can look at the various ways in which we can actually expand that object into some ground object that's completely fully specified. So let's see. We can say that that particular D site that's missing here is free. So that gives us a complete object, a ground object. But we can say, so equally, we would say it's bound to another A. In which case, we're not done yet, because we have to say what's happening on the other side of A. So again, we have two possibilities. Either that other side is free, or that other side is bound to a B. And in that simple universe, that's it. We're done. So we have this ability to manipulate partial objects. And we have to think of partial objects as standing for classes of complete ones. So that buys us the following. We can actually, once we can talk about partial objects, we can talk about partial interactions, partial reactions. So we can, in one sweeping move, write an interaction that's going to stand for an entire class of reactions. Because we have this fine gradedness that's in the syntax to describe the context independence, or the context dependence, conversely, we can do that. So for example, I can say that C binds D if C is bound to that gradient, but maybe C has another side, and I'm not going to talk about that other side, which means that I don't care, which means that I'm assuming that the mechanism which is at play in this interaction is actually insensitive to the presence of this other side and to the status, the binding status, and the internal state status of that other side. So we have the ability to narrow down on a part of the context, and that gives us this combinatorial flexibility and this compactness of expression. So we can go one step further, because we want to model things in a quantitative way, and we can assign to each of our interaction rules, I'm going to call them rules now, a rate. And the rate will be, the average number of times the average number of times one particular event of that type is triggered every time unit of, you choose your time unit. So, what is this? It's a jumping process. The state of the universe is a big side graph. You can think of it as highly liquid, very disconnected. But we know in molecular biology that some of these structures are actually highly connected. Chemotactic clusters of receptors, the postsynaptic density. So it's also possible that the system will be in a phase of matter which is not so liquid. And anyhow, this is the state of the universe. And now we have sitting on top of this microstate, which is every tiny little agent you have in your system of interest. There are the rules. And every rule is actually looking down on the chemical mix, on the soup, and having a certain number of possible matches that will trigger possible events. So we can think of this system, and we can think of the set of the even horizon of the system, the set of all possible events that could happen next. And every one of them will compete with its own rate. So that is what people call in traditional modeling a continuous time Markov chain. And that's this is the operational meaning of writing our rule. And this is where the rates come into play. Now, within all the events of a given interaction rule we're going to pick uniformly at random every single event. And that is the precise counterpart of the assumption that the context does not matter. This is this uniformity in the choice. All right, so let's have a quick example. So we return to our two AB agents. So this is the first rule that says A can dimerize. And this is about the simplest rule you can ever imagine, right? There's absolutely no condition. In particular, I'm saying that the rate at which these two guys bind is completely independent of whether they are already connected to a B or not. Not so for the second rule, where we're saying that B can only bind when the two A's, when that A to which it binds is already bound to another A. So there's a slight condition here. Then the condition will induce some sort of sequencing. If you're a B and you want to bind to an A, then you have to first wait for that A to find another A partner. So this is the beginning. This is the minimal amount of causality. This conditional, this restriction on when a rule can apply. That's going to be used to construct long-range causalities and global behavior. So we can throw in the chemical mix a certain number of A's, a certain number of B's, all disconnected, and we can run our Markov process. So this is one realization. And we have to say what we want to look at. We could look at everything, but here I'm choosing to look at 3 particular observables. And some of them will be actually partial objects, importantly. So B is just a free B. A is just a free A, and here I've got a partial observable, which is the number of AA dimers, such that on one side I yet have to bind a B. So we see that the A's and the B's decrease steadily, because in this simple system there is no way you can detach. So the number of free anything will decrease. But we see that the A's are decreasing faster. And the reason is they have unconditional binding. So this is the queuing effect. The B's have to wait. So this translates into this little time lag. And the green curve, well, the green curve is more interesting. It's not monotonic. We have an increase of dimers, A, of that sort. But then after a while, all the A's finally find their B's. And so this thing is decreasing again. And if you wait sufficiently long, which I didn't do in the simulation, then eventually no object with a free whatever will subsist in the system. OK, so that was our example. So we have this language. We have this stochastic semantics. So now let's see how we can lean on the structure that the language is giving us to describe any particular situation of interest to maybe extract some information about the global behavior. So, beside, I'd like to make a sort of a more conceptual point here. If you picture yourself again the mass, the dense mass of interactions of the systems, then maybe there is no other way to look at the systems, no other way than actually to look at partial objects, right? Because if you're trying to understand, and this is not an operational point of view, it's just a conceptual one, and maybe it's an operational point of view for actual biology, for natural selection. There is no enumeration that will actually deliver any kind of meaningful snapshot of the state of the system. So we have to adjust to this part. This is what makes the systems actually very different than the ones we're used to deal with. OK, so this method that I'm going to present is fragmentation. And eventually what it does, it produces a self-consistent closed differential system that describes the evolution of one particular observable of interest. So there are two ideas. One of them is you pick an observable, and for example, it's going to be the level of activated first free in the model the system we've looked at before. And then you write a differential equation that describes the evolution of this partial object, which we call a fragment, as a function of the concentration of other objects. So now, the second part of the idea is that the partial objects that this differential equation mentions, they are themselves, well, to know the actual value as a function of time, you have to look back at the microstate of the universe. And that's not a good thing, because you'd like to have a closed, self-descriptive system. So what you do is you write a differential equation for those fragments as well. And then you carry on saturating your system of fragments. And each new fragment that's getting in the picture has a causal interpretation in terms of having an interaction with the fragment you started from, the seed, so to speak. And what you get at the end, in practical examples, is a far smaller system than the one you would have gotten if you had done, if possible, a complete ground expansion of the system based on an enumeration of all the possibilities. So the entire process is called fragmentation. So I'm going to describe technically what we're doing here, and then in the conclusion, I'll return to the philosophy of what this algorithm might mean in terms of carrying information. So we'll return to our example, and we look at how we can write this differential system. So we have to make a decision at the beginning. The decision is to have the seed to be a free B in this particular example. So I'm going to call this fragment F1. And now I want to ask, how does F1 evolve as a function of the state of the universe on average? So, well, F1 is a free B. In the system, you can only consume free Bs because there's no way you can dissociate. So there will be only one term, and the only actual rule which does this is the rule number 2 with a rate constant which is K2. And so we have that the consumption of our fragment is proportional to that rate constant and to the number of ways in which you can apply this rule. This is the principle, It's one instance of the mass action principle that's built in the stochastic dynamics. So, you know, in just how many ways can you consume a bee? Well, there's how many bees you have, and there's how many A's you can bind them to. So the result of this is that you're going to have that the time derivative of your fragment of interest is proportional to the number of F1 to the number of F2 as well. So I call this object F2 because it's brought about by rule 2. Now F2 is another partial object that I don't know how to describe the behavior of. So then I have to iterate and I have to ask myself, how do I consume and or produce that F2? So let's see, F2 is that object. So to produce one, obviously, I can use rule R1. So, but I can also consume one by using rule R2. So I've got the exact same negative term. But now I've got this positive contribution. And let's see, how do I produce one more copy of this guy? Well, I have to take an A that's free on the dimerization site, and another one. But one of them at least has to be free also on the B binding site, right? Because this is what this partial object is telling me. So this means that it's going to be proportional both to the number of free A's, F free, and to the number of A's that are free on the dimerization site. So we have a linear expression in F3 and F4 and the rate constant of the rule. And there's a mysterious little combinatorial coefficient here too that I'm going to return to when we have a nice, neat way of describing the underlying combinatorics. So that brings a new pair of partial objects, F3 and F4, which I have now to incorporate into my description. So I'm going to write a differential equation for F3 as well, which is exactly the same term here, but with negative sign. And for F4, I write something else, and then I'm done. So I've got a completely self-predictive differential equation that I extracted starting from my seed. My process is terminated, and I would like now to prove that, one, I can make this a general algorithm, and two, it's actually correct in the sense that the deterministic semantics of that differential system is, in the limit of large populations, an increasingly good, eventually perfect approximation of the behavior of the system I started from. So we can prove it just by experimenting numerically. So here I'm superimposing to the stochastic noisy trajectory the solution of the differential system we've just written. And it's convincing because we see that, for example, this smooth green curve that's generated by the derivative of F2 is actually following from very close the noisy signal that we got from the previous simulation. So it seems that we wrote the correct differential equation. But let's see why. And let's see where the twos are coming from. So this is a piece of technicality, but I thought I would actually indulge into saying this. So let's see how it goes. So what we have is we have this rule R1, and we'd like to, which is the only rule by which we can bind two A's on the dimerization site. And we'd like to answer the following question. What is the rate at which that rule will contribute in the building up of the number of F2s? So it's not a trivial question, because when I pick two A's, because they are partial objects, I don't know whether they're binding a B on the other side. So what I'm going to do, I'm going to look at the even horizon of that rule on my chemical system, on my soup, and I'm going to divide this in three subcases. I'm going to analyze whether the two A's I pick are free on the other side, or whether one of them is free, or whether none of them is free. And then for each of these subcases, I will count the number of F2 I produce per even of that type. By the way, I should mention that you see that the fragments, and this is what makes the wealth of the combinatorial aspects of the system, they are overlapping. They're not mutually exclusive. So that means there's a tiny bit of maths that we'll have to do at some point to make sense of all these numbers. So, but let's do the concrete calculation here. So how many pairs of completely free A's do I have? Well, I have A times A minus 1, because I need to be careful not to pick twice the same, because I cannot self-bind the same, and where A is the number of free A's. So now, if I bind two totally free A's, how many copies of my F2 pattern do I produce? Well, there's a symmetry in this object, so I produce two. My pattern I can actually map one way, the straight way and the twisted way. So this is why the number of F2 productions is 2 for that subset of the even horizon. Now I can pick two A's, and of which only one is free on the other side, in which case I only produce one copy of this pattern. And how many ways in how many ways can I actually do that? When, well, I have to pick one totally free A, one A that's bound on a B, so that's A times B. And again, I can do it in two different ways. So that's my 2AB here. And then finally, I can take two A's that are already bound to a B, but when I plug them together, I don't produce a copy of F2, because F2 needs a B not to be present on A, right? This is what this side is saying here. I'm free. So this means that along my partition, the actual number, the actual yield in terms of producing F2 varies from one subcase to the other. So this means that the stochastic analog of the construction of this deterministic semantics will fail. But I don't care because I'm deterministic, and all I need to compute is the average number of F2 of that uniform population of event, which I'm going to trigger per time unit. So that's proportional to K1, which is the constant, the rate constant of this reaction, and that average, which is, you just compute the average, and at this thing, and we get this term. And this is almost the term that we wrote down in the differential equation, except for a term that is vanishingly small in the limit of large population, which is like the square root of the scale of the system, which is a completely well understood phenomenon that already happens in classical fluid limits of Markov chains. So nothing new here. So that's kind of the pedestrian proof that the differential equation we wrote for that particular system works. But now we can do this on a grander scale for any kind of system. So here is it, briefly. I'm not going to bother you too much with more technical points here, but this is briefly how it goes. We have a reaction, rule, rather. We have a match for that rule. And then we have a fragment that we're going to try to compute the rate of consumption for. And well, for a fragment to be consumed by an event attached to a rule, we need to have a joint embedding of both in the chemical soup in a way that the fragment is mapped to something that's modified by the rule. So here we're constructing a new binding. So this means that we have to look up all possible ways in which we can glue two shapes in this geometric universe. And that's something we can classify by minimal gluings. So this brings us into some well-understood category theoretical or algebraic world where we have to classify co-spans of morphisms using a suitable notion of co-limit. And that's what I'm doing here. So when all is said and done, what we get is the following. We have a generic differential equation that was ID number 1, which splits in two parts, the consumptions with a negative sign and the production with a positive sign. So let's look at the consumptions. For each rule, we have to enumerate the finitely many ways in which the left-hand side of the rule can glue to the fragment of interest. And that is classified by those minimal gluing squares. And we have to compute the concentration in the system X, the state of the universe, of the top of the square, which is the gluing itself. So, and just a slight word of caution here. The map from the square to the top of the square is not going to be injective in general. Actually, we'll see an example. And this is where the combinatorial numbers are coming from, this lack of injectivity. So anyway, so every single contribution here reads like the rule consumes me F at a rate which is, depending on the gluing, proportional to the number of top of minimal gluings into the system. So that's the general principle. Very easy. So the 2 comes from the fact that in this case, for example, we have a production term, which is based on the same principle of counting gluings, but on the other hand side. And here, for example, we have that F2 can be glued to the right-hand side of the rule. So F2, you remember, is this partial pattern we're trying to track the deterministic semantics of. And it can be glued twice because of the symmetry of the right-hand side. And the 2 gluings are actually non-isomorphic. So we have to take both into account if we want to classify carefully the set of events, which is really the generic algebraic trick that we've done in the particular case of splitting the event horizon. So this is how the combinatorial terms come about. So I'm going to skip this. So now there's the other part of the idea, which we have yet to explore better. But one thing we can say is that if the set of completely ground expansions of our system is finite, then the saturation algorithm will terminate. But we have examples where it does not. But these are also examples that have an unbounded number of configurations that are possibly generated by the system. So that's it. So let me skip this. So what's the summary of the algorithm? Well, we can write an exact differential equation for any observable. Each rule will contribute both by left and right gluings. The combinatorics we don't have to be clever about. It's completely packaged by this nice piece of algebra. And to get a closed self-predictive system, we just have to repeat the operation up until it reaches a fixed point. So the projection we obtain, that is to say the deterministic system which is restricted to those fragments that the procedure generated, is an exact projection. So exact in the sense that it fully coincides with the generic ground differential semantics expansion. There's no approximation going on here. And that's a strong contrast with what people are doing when they're doing model reduction. It is irreversible, so I mean, in general, which means that you can't recover the state of the microscopic state of the universe, which makes it actually, I think, more interesting. And it assumes nothing about the parameters at all. It's also scalable. We implemented it using not exactly this procedure, but some approximation derived from static analysis techniques. And we have remarkable compression factors when we apply it to actual rule-based model that have been developed completely independently of the technique. And furthermore, it's going to mesh well with any other kind of approximation. So if you know something about the rates, or if you're ready to give up on negligible observables given numerical information, then you can do that as well. So that's the summary of the technical aspects of the thing. So now the conceptual aspects. I promised that I will return to the big question of how these highly densely connected objects perhaps can deterministically operate at the macroscopic level. So let me do a quick remark first. Mathematically, the deterministic approximation is only valid in the limit of large populations. But large populations of partial objects does not necessarily imply large populations. Sorry, let me say this again. Partial objects can reach the notion of a large population much faster, and in the end with a certain gap compared to the limit of large populations for complete objects, because there's far fewer partial objects than complete objects. So this means technically that we're going to have a convergence theorem that says that the fragmented differential semantics converges much faster than the ground semantics, which is very useful when the ground semantics is infinite dimensional, in which case it doesn't really exist in any proper sense. So fragments can be deterministic where the real system is not deterministic in the traditional sense. So I suggest the following. I suggest that fragments, in this case, that we extract from the structure of the description of the model, might be the variables that you want to track. They're tracking partial objects if you're looking for deterministic behavior. So they might be the basis on which the systems are actually showing reliably a deterministic behavior, despite the fact that everything is highly densely connected and is astonishingly stochastic and drifting all over the state space. So this brings about an entire new set of questions. So these objects, if you think of them as information carriers, are reminiscent of similar situations in statistical physics, and where they are called collective variables. And one of the things that physicists ask in condensed matter physics about collective variables or thermodynamic quantities is that they are robust to variation at the microscopic level. So then it's very natural to ask whether perhaps the fragments are robust to changing the rules. So how robust are they? And then we have to ask, why is the system, if it is, why are the system architectured in this way? And is there any kind of connection we can make to the evolution of the systems in relation to this architecture? So having said that, these are the references, and I'm going to thank you for listening to me. Very good.
So Vincent will take some questions. So at what point will you be able to make an interesting prediction about a protein that would turn out to be true?
Well, wrong predictions are oftentimes very interesting. Yeah. So, and we make lots of wrong predictions.
Right, very good. Well, that's obviously very encouraging. Yes.
So, let me give you an example. So, using these techniques, we can talk about clusters of chemotactic receptors. Mm-hmm. And we can derive by mathematical means conditions on the various parameters in the system for the clusters to be of a sizable dimension. And so when we run our mathematical formula against the actual parameters that we extract from the literature, we find that they are subcritical, which means that the clusters we're actually seeing on the electron microscopy should not exist mathematically. Mm-hmm. So then you're led to say, okay, well, that's a wrong prediction. How can I, what kind of simple mechanism can I assume and re-inject in my description of the system so that I actually get to see what I want to see? And this is obviously some sort of cooperation mechanism will be a good candidate. So that we can compute the minimal cooperativity parameter that we need to do that, and then we can return to experimental people and ask them whether there's any experimental evidence that this is happening. Yeah. Or even go a little further and design experiments that will discriminate between the various assumptions. Mm-hmm. So that's one thing we can do, and that you can't really do with any other syntactic approach.
Please.
So you had a focus on determinism, but these kind of graphical formalisms are often used to talk about other kinds of computational processes that may not even terminate. I was wondering if you've also got some idea on how this might work for some sort of continuous process, and for the properties you might have to talk about of continuous processes.
It's an interesting suggestion, but I would have to think a little further. It's interesting. Are you thinking about any particular? Are you thinking about continuous viable quantum systems? I know Lucas, so I know why you're asking this.
I was actually thinking more about operating systems and about some sort of continuous process that's supposed to have some sort of robustness to a certain amount of interaction, but isn't supposed to do something particular.
I see. Yeah. Well, so these systems actually never really terminate, or if they terminate, the metaphor, the adjustment to the concept of termination you have to make here is that of equilibrium or steady state. And there's an entire set of very exciting questions about whether you can actually inject the physical structures induced by the notion of equilibrium into these kind of languages. So that definitely we are giving very serious thoughts about doing this. That's going to be another lecture, but it's a very good suggestion.
Very good. Please.
Yeah. So the differential equation approach is useful when you have a lot of interacting molecules. What about sort of like cascade type reactions where it just takes like a single molecular trigger to trigger a whole sequence of things? Okay, gradually increasing in magnitude until it has a physical effect.
Right. So well. I don't have a sweeping answer to this. What we provide with this algorithm is we extend the means by the validity of a deterministic differential equation-based description. But nevertheless, as you say rightly, there will still be systems where there's just everything is hinging on just one molecule. And then you have to ask yourself, what is the function behind this kind of system? Maybe, I mean, it's well established that some biological systems actually have an explicitly random design functional behavior, in which case this kind of technique will not teach you anything about the system. So I think it's a fair question. So we extend the validity of a deterministic interpretation of the system, but this is not to say that we make every single system susceptible of this kind of analysis.
Good. And there was another question. Please.
You mentioned that this process is not context dependent. Is that the same as saying it's not path dependent, and that the order that the fragments bind with one another does not have an impact on the long run kind of distribution or state that the system reaches?
Let me see. So I said that the syntax that we're using give you the means to actually express context independence when it happens, when it's in the system you want to describe. If it's not, then there's nothing we can do for you. I mean, but you can actually assume this. So that's one way in which we use the notion of context independence. Now, I think you're also asking another question. Well, you're like kind of asking two questions at once, which is whether the fragmentation algorithm is sensitive to the order in which you do this unfolding. And it's not. It's generic. However, the approximate algorithm that we're using has different properties in this respect. So it's a little of a mixed picture. It's also possible that a fragment will be needed to describe one fragment you started from and not conversely. So you're tracing some sort of causal dependencies when you're doing this thing. I mean, I must say, to be clear, that this research is very recent, as you can see from the references, 2009, 2010, and it's completely an ongoing process. At the time we're speaking, there's lots of people actually trying to improve on this first construction. So we don't understand it very well.
And when there is clear understanding, what will we see? I mean, what would the front cover of Science say? Systems biologists have now.
So a very strong form of what you're suggesting would be to say we have reached a point where we can actually engineer the systems reliably.
All right.
Which is sort of the horizon of synthetic biology, but that's not.
And that would be applied to a particular class of proteins, say?
Yeah, absolutely. And actually, that's right. I mean, if you're doing synthetic biology, and this is a due contrast with systems biology, where you have to actually deal with what you have. In synthetic biology, you're at liberty to choose the components and to re-engineer them so that they're actually more modelable, which is why it's such a tremendous excitement to go into this.
Very good. Okay, now I'll ask Professor Nigel Brown, head of the College of Science and Engineering, to propose a vote of thanks.
Well, I think that was very illustrative of the way that informatics and biology have come together in some areas. And just to tell you a little story, which some of you have heard, systems biology really started in a big way in 2004 in the UK, with government putting significant amounts of money towards it. And when the Comprehensive Spending Review in 2007 came up, the research councils were thinking, well, we can't call it systems biology anymore. You know, we've got to move on. We've got to give it a different name to get money from Treasury. And the Director General of Science and Research then, Sir Keith Nayan, said, no, you've got to keep calling it systems biology. And we looked astonished. And he said, yes, ministers think it's a good thing. We looked even more astonished. And then he said, they think it's changing the behaviour of biologists and computer scientists, so it must be a good thing. And so systems biology has got, you know, considerable funding. And the UK now leads Europe in terms of systems biology. I think we're actually leading parts of the US, certainly, maybe not all of it. And Edinburgh, I would say this, wouldn't I, leads the UK in terms of systems biology. And Vincent and his colleagues in computational aspects of systems biology and in systems biology are one of the reasons that we lead the UK and we lead the Europe. So I'd like you to join me firstly in thanking Vincent in the usual way, and then joining colleagues over the road where I'm told there are drinks and, importantly, canapes. So Vincent, thank you very much for an excellent lecture.
Thank you.
This production is copyright the University of Edinburgh.